We're going to go over cell membranes and transport. So we're really going to focus on how molecules move across our plasma membrane. All right, so the first thing we need to know is that cells maintain a gradient between what's on the inside of their plasma membranes and what is on the outside. And they need to maintain this internal environment that is different from the external environment to regulate the functioning of the cell, to keep in homeostasis. So we would say that we have to regulate a transmembrane gradient. So the transmembrane gradient has to deal with the concentration of solutes. So the concentration of the solutes inside of the cell is going to be different than concentration of solutes outside of the cell. And we can see that in this first image here. We see that the inside is much less concentrated in this particular molecule. Now, cells don't only have to worry about keeping a gradient up with just the molecules, they also have to be aware of any charges that might be associated with the molecule. We would call this the ion electrochemical gradient. So now, instead of just maintaining a gradient of solutes or molecules between the inside and the outside of the cell, now we also have to maintain a charge, right? Now these charges don't have to equal out, but it is going to be a difference in charge. It's going to be a gradient in charge between the inside of the cell and the outside. So if we look at this bottom image here, we're looking at the electrochemical gradient for sodium and chloride. Right? If we look at sodium, which in this image is this purple dot here, we see that inside of the cell, there are fewer of these sodium ions compared to outside. And if we look inside of the cell, if we look at the chloride ions, we see that there are many more inside of the cell than outside. Overall, irregardless of what molecules are inside and outside, looking at just the charges, we can see that inside of our cell is more negatively charged than outside of the cell. Right? So now we have gradients involving ions and gradients involving charges. So the phospholipid bilayer is selectively permeable, which means it's going to allow the passage of some molecules but not others, right? So some molecules can easily diffuse through this phospholipid bilayer, but a molecule that might be hydrophobic, so maybe it's an ion or it has a polarity to it, that's going to be a barrier. They won't be able to easily move through this bilayer. Rather, they're going to need help, and this help might be in the form of transport proteins. Right? But big picture here is that it's selectively permeable. It allows some molecules in and not others, which allows the cell to regulate those concentration gradients, regulate what is inside of the cell versus what is outside of the cell. Now there's different types of membrane transport. We are going to talk about 3 of them here. There is simple diffusion, where substances are moving across a concentration barrier, or concentration gradient. If we look at our phospholipid bilayer here, right, we can see that this solute, this purple solute here, there's many more of them outside or on this side of our membrane than on the other side. Right? And this solution is going to want to move from an area of high concentration to an area of low concentration, which is what it's doing in this image. Right? Now, in this image, it's representing simple diffusion, which means it simply goes through our phospholipids and out the other side. All right? It doesn't need any help. While facilitated diffusion, on the other hand, again, we have this molecule, higher concentration on one side, lower concentration on the other side. It wants to move from a high concentration to a low concentration, and it's doing so, but it's doing so with the help of a protein, or the transport protein. Right? It can't just go through our phospholipids, right? It has to move via this protein. The last type of transport that we will talk about is active transport. And this active transport is where substances are now moving against its gradient. So unlike simple diffusion, unlike facilitated diffusion, where our proteins or our molecules are moving from an area of high concentration to an area of low concentration, right? Now, in active transport, we have the opposite. Now we have molecules moving from an area of low concentration to an area of high concentration. It's moving against that gradient, which means it's going to not only need a protein, a transport protein, but it's also going to use energy, right? It uses up energy when it moves. Right, so passive transport does not need energy. This would be simple diffusion and facilitated diffusion. Active transport, it does need energy. And typically that energy is going to be in the form of ATP. All right, let's look at simple diffusion here. So simple diffusion is where, again, we don't have a protein involved. It can just simply go through the plasma membrane bilayer. But not every molecule can do this. And even those that can go through this bilayer, it might go through, some molecules are going to move through faster or better than others. So this rate of diffusion, this rate of movement, depends on the chemistry of the solute and its concentration. So first of all, it has to be moving from an area of a high concentration to a low concentration. If it's not doing that, it's not going to be able to go through diffusion. Now, other factors that play a part here are the size. Small molecules, they are going to diffuse faster and have the ability to diffuse via simple diffusion. We see in our image here, small molecules might be carbon dioxide or nitrogen or oxygen gases. Right? It could also be things like ethanol, right? Still a small molecule. It does not have a charge associated with it. Some other molecules might have a polarity associated to them. Now, typically if it has a polarity, it's not going to be able to go through simple diffusion, though there are a few exceptions. If the molecule is relatively small, even if it has a polarity, let's say water is a really common example here, it will be able to diffuse across the plasma membrane, but it's going to do so much more slowly. All right, the next factor would be charge, right? So if a molecule does not have a charge and it's relatively small, it can very easily pass across this bilayer. Now, if it has a charge, right, say it's an ion, we look down here at our list, there's this list of ions here, it has these charged, maybe they're molecules with a charge or molecules that are polar. They're not going to be able to get through this phospholipid bilayer. They are not going to be able to go through simple diffusion. We'll see the other methods that they will take here. Right, so simple diffusion, small uncharged molecules are going to work the best for this type of transport. Now, simple diffusion requires no energy. Again, particles are diffusing down their concentration gradient. So if we see in these images here, right, this top image, we have this semi-permeable membrane indicated by these gray dashes, and we have our molecules. Our molecules are slowly diffusing to the other side. And it's important to note that this is not just a one-way thing. Some molecules might get through, other molecules might bounce off and come back. Oop. Once we have a little bit of diffusion going, other molecules might diffuse the opposite direction, right? But overall, in the end, what's going to happen is that we are going to reach this equilibrium. This does not mean that the molecules stop moving, but rather the amount of the molecules on either side of that membrane is equal. There still might be movement back and forth, but it's a balanced movement. We could see the same thing in the second image here, where instead we have two different molecules that are each trying to reach equilibrium by traveling in these two different directions. Right? And again, once we reach equilibrium. It's not like movement stops. Movement is still going to happen, right? But it's balanced out. All right, so let's talk about facilitated diffusion. So facilitated diffusion is a type of diffusion that uses a protein, and it uses a protein called a transport protein. So these transport proteins are essentially channels or passageways for these molecules to get across that plasma membrane. Now, these are really common for ions they are common for ions, so an atom with a charge, and they're really common for any molecules that might be hydrophilic or have a polarity associated with them, right? So if we think about that plasma membrane, the interior is hydrophobic, which means ions and hydrophilic molecules have a really hard time getting through this hydrophobic region. So instead, they're going to take a protein that spans that hydrophobic region, right? It allows them to move through without ever having to interact with these hydrophobic tails. Now we can have two types of transport proteins that we'll talk about here. The first is the channel, the second is a carrier protein. If we look at channels first, channels are an open passageway. So in this image here, we have our transport protein. Essentially, it's like hollow inside. It's like a tunnel, right? If you ever ride your bike on a sidewalk and you have to go under a highway, you might go through a tunnel, right? It's always open, right? So this would be our channel. So ions and molecules that have a charge, or maybe very large molecules, right? Large molecules that can't just simply diffuse, they have to go through a channel instead, are going to use these transport proteins. Now these channels, I mentioned that they're always open. This is not always the case. Sometimes, and most often, these channels are going to be gated, right? So they have the ability to open and close for certain molecules. Once they open, they're like a tunnel, right? But they have a gate, right? So it basically allows for better regulation of which molecules go in and out. All right, the other type of transport protein are transporters, also known as carriers. And these types of proteins, they are not always open. They are not like a tunnel. Instead, they actually change their shape to allow molecules to pass from one side of the membrane to the other. So they go through this conformational change. And if we look at this image here, right, we can see that in this protein, this transport protein, it's open on one side, it accepts a solute, it's going to change its shape to where it empties on the other side, right? So the side that it entered is no longer open, but the side that it wants to go to has now opened up and is releasing the solute on the other side. Right? These types of transporters are really great for larger molecules like sugars, amino acids, or nucleotides, right? Molecules that have charges, molecules that are very large. And so if we look at just how they go through this conformational change, we see that they have these two structural features, right? They have the protein itself, right? But they have this hydrophilic pocket. Now remember, the barrier for a lot of these molecules is this hydrophobic interior of our plasma membrane, right? So we know that they're a hydrophilic molecule that doesn't mix well with the hydrophobic molecule. So therefore, this hydrophilic pocket is very inviting. These solutes will go inside of that pocket, right? And it's going to kind of move like a hinge, right? So it takes it in on one side, and then it releases the solutes on the other side. And it has this ability to go back and forth many, many times. All right, there are different types of transporters. I'm just going to very quickly show you 3 different types. A uniporter, is a transporter that can only move a single molecule or an ion at a time. You see that in this top image here. A symporter can move two or more different ions or different molecules in the same direction at the same time. So here we have a red molecule and this blue triangle molecule, right? They're both entering into this hydrophilic pocket, right, and going in the same direction, being released on the other side. Finally, we have what's called an antiporter. Now, this one also moves two or more different ions or molecules, but they move it in opposite directions. So again, if we look at the image, we see this blue molecule is entering in on one side, being released on the other side. Once on the other side, once this hydrophilic pocket is on the other side, this other type of molecule can bind and move back across the membrane in the opposite direction that the initial molecule did, right? So we have molecules moving in one direction versus molecules moving in two directions, and the number of molecules that can move at a time. Now our last type of transport is active transport. So active transport is different in now that we're moving molecules against their gradient. We are moving molecules from areas of a low concentration to an area of a high concentration. This is opposite from diffusion, right? This is opposite from simple diffusion, from facilitated diffusion, which both of them together would be passive transport because they don't need energy. So we're moving it against its gradient. This is unfavorable, all right? Molecules don't want to move against their gradient. And in order to do so, they need an input of energy, right? So active transport needs energy in the form of ATP. We're going to talk about two different types of active transport, primary and secondary. If we look at active transport, and we're looking at primary active transport, we see, again, we have this protein that spans our plasma membrane. Right? We have a molecule that's moving from an area of a low concentration to a high concentration, so moving against its gradient. And we see that we're using energy, right? So these molecules will go inside of, we call them a pump. These membranes are called pumps, right? We'll go inside of these membranes, these proteins, right? And then be released to the other side. But in order for it to make that conformational change, it needs ATP. It needs to have some energy in order for it to change the shape. Now, secondary transport is a little different, right? So secondary transport, we're still moving molecules against their gradient, but we are using a different way, right? We're essentially using a pre-existing gradient to drive transport instead of directly using ATP. So, primary active transport, we've used energy, we've built up this gradient of these white triangle molecules here, and now we're using this built up gradient, because molecules don't want to go against their gradient, they want to go with their gradient, right? So we're using that to power our secondary active transport. Right? So we used ATP in the first step, where we built up this gradient. Molecules want to move from an area of high to low, right? So now that we've taken this molecule, moved it against its gradient, it's going to want to go with its gradient, right? How molecules want to move. And so this molecule enter into this symporter, right? Will enter into it. And once it enters in, it kind of gives a signal for the conformation change to happen, right? But this is a symporter. We're transporting two molecules at the same time. So the other molecule that's being transported is the molecule that's going against its gradient, right? This purple molecule here. You see it's going from an area of low to an area of high. Versus this triangle molecule, is now going from an area of high to an area of low, right? So we're using this triangle gradient, right? This molecule wants to move to the other side, but it's going to take along a molecule going against its gradient, right? Once both are in place, then the symporter will open. So it doesn't directly use ATP, but in order for that gradient to be built up in the first place, ATP had to be used for one of the molecules. All right, so we went over 3 types of transport here. Passive transport requires no energy. Active transport needs energy, right? Diffusion, facilitated diffusion, so whether or not you use a protein embedded in that plasma membrane can be passive transport, right? We're going from an area of high concentration to an area of low. Osmosis, which we'll talk about next in our next lecture here, is also a type of passive transport because it also does not require any energy. And then we have active transport. It requires energy and we're moving a molecule against its gradient. All right, so this is a nice review slide here. So what determines how a molecule or atom can cross the plasma membrane? Right? I'm not going to read out the entirety of this slide here, but big picture, it depends on if you have a polarity or if you have a charge. It depends on your size. And it depends on the gradient. Right? So if you have a charge or a polarity, more than likely, if you're going with your gradient, you're going to be using facilitated diffusion. If you're a very small molecule or a molecule without a charge, you can likely use simple diffusion. And if you're a molecule, no matter the size, right, if you're moving against your gradient, you're going to have to use active transport. All right, so in our next lecture, again, we'll elaborate on osmosis, but we will also practice these different types of transport across our plasma membrane.